First of all, thank you everyone for taking this seminar. As Jinsu mentioned, the first topic of this seminar will be the one-stop solution for the ADC discoveries. Let's begin with a very brief overview of the ADC landscape. So this table summarizes the so far 7 launched ADC products for blood tumor, the hematological malignancies. Most of these drugs are approved before the year of 2020, and they are approved for most of them are approved for the indications including like myelomas, leukemia. So this is for the first part, and the second table summarizes the 8 drugs currently marketed for the solid tumors, and the majority of them are approved after the year of 2019. As you may see from these tables, the solid tumor is gaining more and more interest in ADC markets in the recent years. As you may well know, the ADC is comprised by 3 parts: the naked antibodies, the payload, and the linkers. For the discovery of the naked antibodies, usually we will focus on the profiling of 6 different aspects, including the antigen binding specificities, affinities, the cross-binding to cyno-monkey antigens, the internalization efficacies, the biophysical, biochemical properties, which is developabilities, and the immunogenicities. In addition to the naked antibodies, the selection of the payload and linkers has to be made based on your target MOAs, the target biology, the disease biology, including the toxicities, the hydrophobicities, the PK profiles, and etc. In order to give a comprehensive profiling of your ADC products, after the conjugation of the ADC drugs, usually we will evaluate the candidates from different perspectives, including in vitro assays, in vivo disease models, and also the general developabilities. The discovery of ADCs workflows can be categorized into 7 stages. The first stage, the target discovery. Usually this is a time to define your target product profile, the TPP, based on your target biology, the disease biology, and the indications, and also the competitive landscape. Once the TPP is defined, we will move on to the second part, the naked antibody discoveries. People commonly use a variety of platform technologies like hybridomas, phage displays, and the single B cell to generate antibody leads for the ADC candidates. And during this stage, the assessment of internalization is a very crucial step to narrow down your candidates for the ADC applications. At the same time, we'll also start to think about the selection of a payload and linkers. Once both parts, the naked antibody, payload, and linker are decided, we'll move on to the stage 4, the ADC conjugations, using technologies like splicing or cysteine-based semi-random conjugations or the site-specific conjugation technologies to link the payload to the naked antibody carriers. Following the conjugations, we will evaluate the ADC candidate by in vitro assays, in vivo disease models, and also the developability analysis for stability assessment. Okay, next, let's start with the naked antibody discoveries for a more detailed discussions with the workflows we just introduced in the previous slide. So far, the technologies used for naked antibody generations can be categorized as 2 major types: the animal immunization-based, or called the in vivo-based approach, and the in vitro-based approach. Both in vivo and the in vitro approach can be achieved by platform technologies like hybridomas, a single B cell, and phage displays. And the final leads can come in as the formats like traditional full-length IgGs or antibody fragments, including Fab, single domains, or single-chain FVs. Among these different technologies, hybridoma is the one we recommended for the specific ADC applications, the screening of antibodies for ADCs, because using this hybridoma-based platform, or we call the powerdoma, you can use the hybridoma supernatant to conduct the high-throughput internalization assays at a very early stage. These particular features can significantly improve your project's success rates and reduce the overall timeline and the cost. So basically, it can help you narrow down the antibody binder candidates based on the internalization kinetics, which is a necessary indicators for the ADC candidate. Speaking of the internalization, currently there are 4 different approaches to quantify or to measure the internalizations, which is the live cell imaging systems, the toxin conjugated approach, the pH indicator, the pH rodel, and the temperature shift-based assays. The top 2 ones comes with the high throughput and automatic platforms. So these tools are the ones we usually recommend it for discovery stage for the purpose of early stage high throughput screening, because it can run through hundreds, even thousands of samples and provide much better signal sensitivities and assay robustness. Let's go over these 2 different assays one by one. First with the live cell imaging systems. So this assays is achieved by a hardware called InQSight. I believe some of you guys are already familiar with it. So the net rationale of this hardware is to utilize a pH-sensitive fluorescence dye. And when the antibody take this dye into the cell, into the lysosome, the acidic conditions in the lysosome will trigger the fluorescence dye and make it start to glow. You can start to, and the InQSight will start to collect the fluorescence signals. The intensity of the fluorescence signals is proportional to the rate of internalizations. So basically, this is how we determine the internalization based on the fluorescence signal intensities. The key feature of this platform is able to evaluate, to measure the internalization at 2 different levels. Traditionally, people do this kind of assays by a dose response curves, like the graph to the bottom right. So basically, we can establish a correlation between the antibody concentrations on the axis and the internalization intensities on the y-axis. So this is a typical dose response curves. In addition to this, this InQSight live cell imaging system can also provide a time course based analysis. On the x-axis, it's a scale by hours, and on the y-axis, it's the intensity of internalization. Basically, this analysis can provide the kinetic data of the each antibody internalization on a specific cells on a specific time point. Alternatively, we can use another high throughput system. It's called DT3C conjugation systems. So DT3C is a synthetic toxins. It's not very commonly used for ADCs, but it is commonly used as a reagent or as a tool to measure the internalization. Similar to the pH-sensitive dye, this toxin can associate with antibody constant region non-covalently. So basically, you do not need to do any chemical reaction to conjugate the toxin to the antibodies. It's a simple mixture and incubation process. So after the incubation, the antibody will take the toxin into the cell and start to kill the cells. By the measurement of the cell death, we can convert the cell death to the relative internalizations. And the key feature of this platform is it's the high throughput systems. You can do it in either 96 well plates or 384 well plates. It's pretty similar to the traditional Fab-Zap systems. The only difference is it provides much higher throughput. So it's particularly useful when it comes to earlier stage, larger scale binder screenings. Once the naked antibody is determined, the candidate is determined, we'll move on to the ADC pharmacology. Start with the payload linker conjugations. So as I mentioned at the very beginning, the selection of the payload and the linker has to be made based on the MOAs. And the ProBio offers a large libraries, including more than 150 cytotoxins and more than 950 linkers to select from to choose the right combinations for the ADCs. And in terms of the conjugations, ProBio provides the traditional cysteine-lysine-based semi-random conjugations and also the site-specific glycan side chain-based conjugations. I will share a case studies to compare these two different conjugation methods with a GPRC5D case studies in the large part. So after the conjugations, the first bioassay, the cell-based assays, is the most traditional cell-killing assays. The cell killing can be measured by either like a LDH or CTGs or CCK8. In addition to cell viability assays, you can also do the cell apoptosis assays or cell cycle assays to evaluate the drug efficacies. In addition to the cell killings, bystander effect is another crucial experiment for some of the ADCs. These can be achieved by the same machines I mentioned at the very beginning for the internalization, the InQ site. Basically, in these assays, the HER2 positive cells are labeled with a red fluorescence, and the HER2 minus cells are labeled with a green fluorescence. Both cells are incubated together along with your ADC candidates. And the InQ site will measure the cell death by the signal intensity of a green or red fluorescence. And the decreasing signal of these fluorescence is correlated with the cell death. And if you see the signal reduce with the green fluorescence, that means the ADC has a significant bystander effect, which can kill the HER2 minus cells after entering the HER2 positive cells and releasing from the HER2 positive cells. So after these two typical cell-based assays, we'll move on to the next part, the in vivo studies. It's a pretty standard oncology studies, including the efficacy, the tumor model studies, the PK study, including both in ex vivos and in vivo PK studies, and also the toxicity studies. For ADCs, the toxicity studies is pretty crucial because most of the ADCs has these toxicity issues, the risk. So that's why we normally recommend you to conduct the early stage of toxicity studies at the discovery stage using either rodents or a non-human primate. To conduct the PK and AD studies, you need a panel of reagent antibodies like anti-EDO type antibodies. And the generation of anti-EDO type antibodies could be very time consuming. Most likely, you will probably need 5 to 6 months to generate these reagent antibodies to develop the PK and AD assays. ProBio offers a panel of off-the-shelf, ready-to-use anti-EDO type antibodies specific for the naked antibody part and the small molecule parts, so it can be easily paired with each other for the purpose of PK and ADA studies. So once the initial ADC candidate is characterized and finalized, we will move on back to the naked antibodies for engineering and the final developability assessment. Humanization is a pretty common antibody engineering approach nowadays. It's really quick, only takes about 3 weeks, and usually it comes with the guarantee of affinities. Affinity tuning is an optional process depending on the antibodies, the affinities of your parental antibodies. If you want to increase the affinity and to increase the toxicities, you can do affinity maturations. Alternatively, if you want to decrease the toxicities, you can do affinity attenuations to decrease the affinities. The third part, FC engineering, is not a very popular topic in terms of for ADC discoveries, because so far most of the approved ADC drugs use the wild-type ADCs. However, some people may prefer using FC enhancement, enhanced FC effector mutations to add ADCC or CDC cytotoxicity to the ADC toxin-based cytotoxicities. Alternatively, some people believe that the FC-mediated macrophage internalization could be a possible reason for non-specific toxicity of ADCs. So in that circumstance, people may want to mutate the FC regions to silence the FC effector function to avoid the FC-mediated internalization, like the phagocytosis. Once the antibody sequence is finalized, we'll move on to the developability assessment. Both the naked antibody and the conjugated ADCs needs to be assessed for its stabilities under different kinds of stressful conditions, including like high temperatures, repeated freeze-thaw cycles, and acidic conditions. Okay, so for the last part, let's have a review for case studies. So this case study is a discovery of ADCs against GPRC5Ds. GPRC5Ds is a very popular tumor-associated antigens in recent years in both cell therapies, in T-cell engagers, and also in ADCs. It is a typical 7-transmembrane GPCR proteins, which is considered as a really challenging target because most of the time you cannot produce high-quality recombinant proteins for the animal immunization to generate antibodies. So that's why people often rely on DNA immunization or whole cell immunization to generate antibodies against the GPCR targets. However, DNA and cell immunization is relatively low efficient in terms of the generating antibody binders to these multiple transmembrane targets. So as a result, in these case studies, we use mRNA instead of DNA and whole cell to immunize the mice for the antibody generations. So after the hybridoma fusions, we screen the hybridomas with both engineered overexpressed human and cyno GPRC5D cell lines, and also the myeloma MM.1S cancer cell lines for confirmation of actual cell surface bindings. In addition to the antigen bindings, we also screen through thousands of binders with the internalizations, the one I mentioned previously, using either the incucytes or the DT3C systems to narrow down the candidate to roughly 50 hits. And these hits were subcloned and confirmed by binding and internalization. After sequencing, we narrowed these candidates down to 21 unique leads. And based on the sequence analysis, we selected 7 of them for further characterization and payload conjugation and also the ADC pharmacology characterizations. Next, I'm going to share a series of detailed data for these case studies. First of all, it's the efficiency of the mRNA immunizations. So the purpose of this study is to compare the efficiency between the mRNA versus the DNA plus whole cell immunization. As I mentioned previously, the DNA and whole cell immunization is considered relatively lower efficient in terms of generating antibody binders. So in this experiment, we compare these 2 different kind of antigens at 3 different levels. The first is by in vitro transient transfection of mRNA versus the DNA. The light green is for the mRNA, the dark green is for the DNAs. And we use 3 different reference antibodies to quantify the cell surface expression level of a GPRC5D transmembrane proteins. As you can see from this data, the mRNA can express the GPRC5D at a much higher level on the cell surface comparing to the DNA in the in vitro transient systems. And the second step, we compare these 2 different kinds of antigens during the immunization stage by comparing the animal serum titer data. The calculation of the titer is determined by the ratio of flow cytometry intensities, the MFI values, on the antigen positive cell line versus the antigen negative cell lines. So that's why it is a ratio. The mRNA immunization after 3 rounds of mRNA immunization, it will create a cell titer ratio at more than 600, while the DNA plus cell immunization can only generate a cell titer ratio at about 4-fold. So there is like at least 150-fold differences between these 2 different kind of antigens. So this further demonstrate that the capabilities of mRNA to induce the immune responses to generate much more antibodies comparing to the DNA and the cell approach. The third level on the right panel is basically by counting the total number of positive antibodies after hybridoma screenings within different affinity range. The affinity range is calculated with a similar approach as the serum titration data. Basically, we compare the flow cytometry MFI value on the antigen positive cell line versus the antigen negative cell lines to calculate the ratio. And we have a different range of the ratio here, 1 to 5, 5 to 10, 10 to 20, 20 to 50, and above 50s. And within each range, we counted the total number of positive binders. The top 2 rows are the animals immunized with mRNA, and the bottom 2 rows are the animals immunized with DNA and cells. So based on the number here, you can see the mRNA is able to generate a lot more, hundreds of more positive binders comparing to the DNA and the plus cell immunizations. So after the screening process, as I mentioned in the previous slides, we selected the final top 7 candidates. And first, we evaluated the affinities, the cell binding affinities using overexpressed engineered cell, the myeloma cancer cell, the MM.1S cell line, and the transient expressed cell with the expression of a Sinus GPRC5Ds. In all of these 3 experiments, we see comparable or better cell binding affinities from these 7 candidates comparing to the clinical benchmark. In addition to the antigen binding affinities, we also evaluate the internalizations with these naked antibodies using both InQSite at 24 hours and the DT3Cs at 72 hours. So these assays are done against the GPRC5D overexpression engineered cell lines. As you can see from these two figures, all of these 7 candidates shows similar equivalent or better internalizations comparing to the clinical benchmark, both of these two clinical benchmark in these two independent systems. In terms of internalization, in addition to the engineered cell lines, we also did it with the myeloma cancer cell lines, the MM.1S. So since the GPRC5D antigen densities on these cancer cell lines is significantly lower than the overexpression engineered cell line in previous slides, so the overall signal you are seeing in this page is relatively lower than previous page. But however, we're still able to see a robust internalization of these 7 candidates, which is equivalent to the clinical benchmark. So after careful characterization of naked antibodies, next we'll move on to the evaluation of the ADC pharmacologies after the conjugation with 2 different approach. Here, the G stands for the glycan side chain based site-specific conjugation, while the P stands for the cysteine based semi-random conjugations. So both sets of samples, we analyzed them with in vitro assays and in vivo disease model, the tumor models. So there's a lot of data in these figures. It's just too crowded to differentiate from one samples to another. So we separate them into 6 different groups. In each group, it's the same ADC candidate conjugated with 2 different conjugation approach. The P stands for the cysteine, cysteine based semi-random conjugation, and the G stands for the glycan side chain based site specific conjugations. And first, we evaluate the distribution of species of the ADCs in different DAR range from 0 to 8, to DAR 0 to DAR 8. As you can see from the top left tables, with the cysteine-based conjugation approach, you see a various distribution of the ADC product. The DAR4 takes about 43% of the total species, but there are significant amount of ADCs distributed on the DAR 0, DAR 2, DAR 6, and the DAR 8. The average DAR is about 3.4. However, when we switch to the glycan-based site-specific conjugations, the DAR4 species takes about 83 to 84%, and the remaining are the DAR2 species. So this data suggests a much better homogeneity with the site-specific conjugations comparing to the traditional cysteine-based semi-random conjugations. Next, we take these 2 samples for further pharmacology assessment, including cell-based assays in the middle panels and animal-based tumor model analysis on the right panels. So surprisingly, in the cell-based assays, the cysteine-based conjugation approach gives a better killing efficacies comparing to the glycan-based site-specific conjugations, as you see in this middle graph, the green curve versus the blue curves. This is a little bit of a surprise to us, but in the disease models, in the tumor models, we see the reverse patterns. The site-specific conjugated samples shows a much better tumor inhibition, which is red, comparing to the blue one, which is the semi-random, the cysteine-based conjugated samples. And we'll move on to the next samples, the clone number 53, the same two different conjugation approach, the same distribution analysis for the DAR distributions. We see similar patterns. For the site-specific conjugations, almost 80% of them are DAR4s, but for the cysteine-based conjugation approach, only 43 of them are DAR4s. And again, in the cell-based assays, we see these two drugs showed similar efficacies. However, the semi-random conjugated samples shows a little bit better Emax, but a little bit higher EC50s. But when we switch to the tumor models, the in vivo studies, again, we see a similar patterns like the first candidates. The site-specific conjugated samples, the brown one shows much better tumor growth inhibitions comparing to the cysteine-based conjugated samples. The similar profilings, we also seen similar profilings with another several candidates, the clone 52 and the clone 402 and the clone 390s. Similar or worse in vitro killing from the site-specific conjugated samples, but much better tumor inhibitions in vivo models. But out of these 6 samples, the clone 411 showed a reverse patterns. The cell-based killing efficacy is pretty much similar, but this time the site-specific conjugated samples showed a worse tumor growth inhibitions comparing to the cysteine-based semi-random conjugated samples. So based on the data, let's first look at the cell-based killing assays. Although the site-specific conjugations can generate samples with much better homogeneities, but our theory or our guess is those high DAR species generated from the random conjugation methods has a better potencies in terms of the cell killings. That can explain why we see better cell killings with those cysteine-based conjugated samples. However, in the in vivo animal models, 5 out of the 6 samples showed better tumor growth inhibitions with the site-specific conjugations. So we believe that the homogeneity of the samples may have a bigger roles in actual tumor tissues. In addition to the homogeneity, the antibody itself may have certain functions in terms of these discrepancies of in vivo tumor growth between the site-specific conjugations versus the cysteine-based conjugations. Okay, so that's pretty much everything for my talk. Let's move on to the Q&A sessions. Any questions?
네, 혹시 질문 있으시면은 시간 관계상 먼저 한 개만 받도록 하겠습니다. 네, 따로 질문 없으시면은 첫 번째 섹션은 여기서 종료하도록 하겠고요. 혹시라도 질문 있으시면은 저희 이제 브레이킹 타임을 좀 이용해 주시면 감사하겠습니다. Thank you, Derek.
Okay.